Hi everyone, welcome to my channel Microbiology with Sumi. Please like, share and subscribe to my channel. Today we are going to give a look upon design of fermentation media. Let's start with introduction. First point is fermentation is a metabolic process carried out by some microorganisms in which sugar is consumed in absence of oxygen and economically important byproducts are given out. So here a microorganism carry out fermentation in absence of oxygen, and as a result they give out economically important products. Second, in fermentation, the most important things are fermentor, fermentation media, fermentation organism and optimum condition. So these are the four important basic things that are required for carrying out a fermentation process. Further, optimum conditions and desirable fermentation media are very important for obtaining high yield of product. If we provide optimum conditions and highly nutritional fermentation media to microorganism, they will definitely give us high yield of product. The quality of fermentation media is important for growth of microbes. Fermentation media provides substrate for synthesis of product in fermentation process. So when we provide a substrate, they will ultimately give us a product. So good quality of fermentation media, that is good quality of substrate, is very important for obtaining a good quality of product in fermentation process. Let's study some important points about fermentation media. Fermentation media consists of two important components, that is major components and minor components. Major components are carbon source and nitrogen source, whereas minor components are inorganic salts, vitamins, growth factors, antifoaming agents, buffers, dissolved oxygen, other dissolved gases, growth inhibitors and enzymes. Further, requirement of nutrients depends upon type of organism used and type of fermentation that is carried out. So the choice of nutrients that we are going to supply to the microorganism, it depends upon two things. First, the fermentation organisms which we are using, and second, the type of fermentation we are carrying out. Further, types of nutrients present in fermentation media determine the yield of product. Now here the type of nutrients means we can say what quality of nutrients, which nutrients we are going to supply to the organisms. It completely determine the yield of product. Means the substrate or the nutrients which we are providing to organism will result in the yield of product. There are two uses of fermentation media. First, growth media, that is, it contains less nutrients and used for growth of microorganism and preparation of inoculum for fermentation. So there are basically two types of fermentation media. First is growth media and second is fermentation media. So first, growth media, it is completely used for only growth of microorganism and preparation of inoculum, that is the amount or the culture which we are going to add in the fermentation. So this is the role of growth media. Second is fermentation media. Now fermentation media, it contains large amount of nutrients. This media is used in fermentation industry for production of economically important product. Now fermentation media is simply the media that is used for fermentation and from which the microorganisms are going to use the nutrients and give a economically important product, that is a fermented product. Role of fermentation media. During fermentation process, organisms enjoy luxurious metabolism with high amount of nutrients and optimum conditions. So when a fermentation process is carried out, these organisms are exposed to high amount of nutrients, that too in controlled optimum conditions. So in this situation, these organisms enjoy their luxurious metabolism. Further, due to presence of large amount of nutrients, the microorganisms become hyperactive and consume excess nutrient and result in partial degradation of fermentation media. So this is normal. When a microorganism is exposed to a large amount of nutrient, that microorganism becomes greedy and hyperactive. As a result, it tries to consume as many as nutrients as it can, and this results in partial degradation of fermentation media. Further, the waste product excreted by microbes may be the desired product. Now here they are going to consume large amount of nutrients, and as they consume so many nutrients, they are going to excrete something, and this excreted product may be our desired product. Further, the substrate given to microbes should not reach the inhibitory concentration level, as it may result in death of cell. Of course, here in fermentation we are going to provide them luxurious metabolism, but this amount of nutrients which we are going to provide them, they also have a limit. This limit should not be exceeded beyond inhibitory concentration level, and which may result in death of cell. Now what may happen if we provide excess substrate? It inhibits vital enzymes and may result in death of cells. Also, the water which is present in cytoplasm plays an important role for metabolic process. If excess sugar or salt is available in this fermentation media, it would tie up the cytoplasmic water, and which result in lack of water for metabolism and cause death of microbes, and this may affect our fermentation, and industry can face a loss. Further, see, water present in cell cytoplasm is an important factor, as I told before. Excess substrate may increase osmotic pressure and affect enzyme activities in cell. So if the substrate is provided in extra amount, it may increase the osmotic pressure on cell, and due to which the enzyme activity, which is very important in fermentation, can affect, and which result in damage of our fermentation process. Further, microbes excrete this excess substrate in the form of partially digested fermentation media. Now let's see types of fermentation media. There are basically two types of fermentation media. The first is synthetic media and the second is crude media. Let's give a look upon synthetic media. The first point is the exact composition of nutrient is predetermined. That is, the exact composition or the nutrients which are going to be added in synthetic media is predetermined. It is known. Second, here level and concentration of nutrients can be controlled and media can be redesigned. As we know, each and every nutrient that is going to be added in the media, the amount or the concentration of nutrients that we are going to add in this media, so we can adjust the level and concentration of nutrients, as well as we can redesign any media as per our demand or requirement. Further, used in experiment related to effect of nutrients on growth of microbes and yield of product. Now synthetic media is generally used for experiment. Now what kind of experiment? The experiment includes the result or the effect of nutrients that on the growth of microbes as well as yield of product. So which suppose for example, if we add this so and so nutrient in the media, what will be the yield of product? We can experiment certain things by using synthetic media. Further, metabolic pathways and main ingredients that is involved in product formation can be determined. As I told, we can determine metabolic pathways as well as the ingredient which is responsible for the product formation, or by which ingredient we can get high quality of product or more yield of that product can be determined. Further, synthetic media can be optimized as per our requirement. As I told before, we can adjust the level of nutrients, the concentration of nutrients as per our requirement. Product recovery is easy as all product pure ingredients are used. So when a fermentation is carried out, or when a experiment is carried out, the product which we obtain can be recovered easily, because all ingredients we are using in synthetic media are known to us and they are in pure form. Further, it is expensive media and cannot be used on industrial scale. As a process, it is not economical and profitable. As I told you, we are going to use pure ingredients in this media, and pure ingredients are very costly. So this is not profitable at all on industrial scale. We can only use it for our experimental work as well as. Let's see the next media, that is crude media. Crude media contains a rough composition of media, that is we don't know the ingredients, the nutrients or the substrate which is going to be used in the media. It gives high yield of product and contains undefined source of ingredients. So as I told you, we don't know the ingredients, the nutrients that are going to be used here, but it gives a high yield of product. Further, it contains high level of nutrients, vitamins, proteins, growth factors, anti-foaming agents and precursors. The last point, it is important to ensure that crude media should not contain any toxic substances. So it is very important to determine whether the media contains any toxic substances or it doesn't contain any toxic substances. If it contains toxic substances, it may harm the microorganisms and which may cause economic loss in fermentation process. Let's see the important ingredients of crude media. The first is inorganic nutrients, carbon source, nitrogen source, growth factors, precursors and buffers. Let's study the crude media components one by one. The first is inorganic nutrients. It contains salt containing anions and cations along with carbon source specific ion like magnesium ion, phosphate and sulfates are added to balance crude media. Second is carbon source. Simple to complex carbohydrates are used. Different sugars, organic acid, fatty acids, proteins, peptides are added as a carbon source. Carbon source is the most important ingredient in any fermentation process. So in fermentation media, crude source of carbon is added. Now what are the examples of carbon source that are added in crude media are blackstrap molasses, corn molasses, beet molasses, starch, corn, rice, rye, milo, wheat, straw, wood waste, sawmill, etc. These all are the sources of carbon that are added in the crude media. Further, nitrogen source. Now, non-proteolytic microorganisms need pure form of urea, ammonia and nitrate as a source of nitrogen. After carbon, nitrogen is also very important in fermentation media. When fermentation organism is proteolytic in nature, animal or plant raw material are also used as a nitrogen source. The examples of nitrogen source are distillery, dried solubles, casein, cereal grains, peptones, yeast extract, hydrolysate and soybean meal. The fourth factor is growth factor. Crude media constituents provide enough amount of growth factors, so no extra addition of growth factor is required. If there is a lack of any kind of vitamins or nutrients, growth factors can be added to media. Now the examples of growth factors are yeast extract and beef extract. Further, precursors. It is a crude constituent. Precursor incorporate in the molecule of product without changing the final product. It improves the yield and quality of product. In short, this is also important point. Example of precursors are cobalt chloride is added in fermentation of vitamin B12. Now the last is buffers. Buffers are used to control drastic change of pH. Sometimes media components may act as buffer. Now buffer maintaining the pH is one of the important optimum conditions, and these buffers maintain the pH to the optimum levels. For example, protein, peptides, amino acids act as a good buffer at neutral pH. Sometimes inorganic buffers like K2HPO4, KH2PO4 and CaCO3 can be added as required. Generally, during the fermentation process, pH changes to acidic or alkaline pH, and the cheapest and easily available buffer is CaCO3. This was all about. Thank you for watching my video. If you like my video, please like, share and subscribe to my channel. Thank you.